Hello, this is Michael Lawless, and in this video I'll discuss metabolism predictions in ADMET Predictor. This slide shows drug metabolizing enzymes. It's from a publication in Drug Metabolism and Disposition in 2004, and it shows the enzymes metabolizing the top 200 compounds. So here we have about 3/4 of them are metabolized by CYP. It's hard to say, a little less than that obviously for UGTs, and a little bit of that for esterases. Then of the CYPs, it's broken up into the 5 major metabolizing enzymes: 1A2, 2C9, 2C19, 2D6, and 3A4, with less contributions from 2B6 and 2E1. Also an emerging enzyme for drugs is aldehyde oxidase, and this can metabolize some of the newer kinase inhibiting drugs. ADMET Predictor can predict CYP and AOX metabolites, as well as esterase and UGT metabolites. And I'll discuss each one of those separately. First, I'll discuss our models to predict CYP metabolites. Here I've just taken a single molecule, and I'm going to click on the Add Me button and then calculate all ADMET properties. Then I'll move over to the Metabolism tab here. And the first thing we see are the CYP substrate, non-substrate star plots for this particular compound. And if we hover over it, we see that it's predicted to be a 2C9 and 2A6 and a 2C8 substrate. It's evaluated all the models for the 9 isoforms of cytochrome P450. If it predicts no, such as this prediction for CYP 1A2, then it doesn't predict sites, KM, Vmax, clearance, etc. So let's pin this column here and then walk over to the first substrate model where it predicts yes, which is 2A6 here. Now note that it predicts yes, but the confidence isn't very high. You've got to assume that it's kind of undecided here, and if you really wanted to find out if this was a 2A substrate, then you need to perform some experiments. So that's a little bit iffy prediction, but since it did predict yes, then in this next column we have the sites of metabolism. So these are the 2 atoms that it's predicting to be metabolized, atom 15 and atom 11. And if we simply click on the cell, then it brings up this dialog box here where it's marked the 2 atoms in here. One is the 15 and the other corresponds to C11. And we can put the structure numbers up here by holding the control button and then clicking on atom numbers here. So we see 11 and 15 are the sites of metabolism. And then these are the scores. So this our sites of metabolism models are actually classification models built with atomic descriptors, where each row in the QSAR table is going to be an atom in the molecule. So it would produce a row for each one of the atoms here that can be metabolized. So this oxygen and these oxygens are not considered CYP sites of metabolism because CYPs don't metabolize oxygens. There needs to be a carbon with a hydrogen on it. It can also be a nitrogen or a sulfur for the N oxide and the S oxide. Then within the, we develop the model, and these are the scores or the outputs of the model. And the default is if a compound is predicted to be a substrate, then the highest scoring atom from the sites of metabolism model is marked as a site of metabolism. There's also a cutoff on what score can also be marked as a site of metabolism. So here it's this atom here. And note that this has a high score and this has a high score, but it didn't quite make the cutoff. So you may want to look at those scores later on. And then similarly, we're going to have similar ones for 2C9 and 2C8. Now if I double click on the molecule and click on the MET button, you'll see that it predicts these 2C9 sites of metabolism. And here there's 3 for it. And so it simply hydroxylates them. Going back over the procedure, first it predicts if it's a substrate or non-substrate for a certain isoform of cytochrome P450. If it predicts a substrate, then it predicts the sites of metabolism. Now once it's predicted the sites of metabolism, we go into MedChem Designer it will pick up that there's 3 sites for this particular isoform and then insert an oxygen between the CH bond. And this is done through some files called CRF files, and I'll explain those in a little bit. But essentially, it obtains what atoms are marked and then uses a smirk string to transform the molecule into the product. Now note we don't see depictions for the other 2 enzymes, and that's because the default is only to display the 5 major isoforms. Let's go into the settings dialog box to change the default behavior of the CYP metabolites. So I bring up the change settings dialog box, click on the metabolite tab, and then just check all of the CYP isoforms here. Click OK, and then when I generate metabolites, now we see metabolites corresponding to 2C8 and 2A6 here. For this top metabolite, 2C8, you see that it's cleaved this bond in here. So it's hydroxylated this position. It's an unstable intermediate. So it cleaves this bond to form the ketone and the sulfonamide. We can visualize that metabolite by again going back into the settings dialog box under the metabolites tab and switching the observe metabolic intermediates to all. We'll click OK and then use the window to go back to the original molecule. Now when we generate sites of metabolism, you see we have shown the intermediate here. Sometimes, our transformations are wrong, so that actually this is a stable intermediate and can be isolated in the experiment. And that's why we want to show the intermediates here. Here I want to show an example of kind of an unusual transformation. So we have a agrochemical. I'll double click on this and then just simply click on the Met button here. And I want to discuss this particular metabolite here, which takes this sulfur and converts it into an oxygen. So this is kind of a known transformation for this class of agrochemicals. And we've actually included that and marked this as a site of metabolism. And then the transformation rules that I mentioned earlier convert the sulfur into an oxygen. In this next section, I'll describe our aldehyde oxidase. These models work in the same way as the cytochrome P450 models in that there is an AOX substrate non-substrate model. If it's predicted to be a substrate, then we predict the sites of metabolism for the compound. This example compound is BIBX. It's an EGFR kinase inhibitor, and it had a very low oral bioavailability. And they found out that this was due to aldehyde oxidase metabolizing the compound. If I take the compound and then calculate ADMET properties, then I'll go over into the metabolism tab and then scroll over to the right to find the AOX models. So we have a substrate, non-substrate. It's predicted yes for that. Then if we double click on this cell, then we display the 2 sites predicted for AOX metabolism here. We can double click on the molecule to bring it up into MedChem Designer. And then I'm going to go into the settings here and turn off everything except the AOX enzyme. Then click OK, and then just generate metabolites. And you'll see that it first hydroxylates this position, so that's what AOX does, and then that rearranges into the ketone. Similarly, we hydroxylate this position, and then if we flip this bond over here and bring the NH over, the hydrogen over here, we form this tautomer. In this next section, I'll describe our UGT models. These are a little bit different than the CYP and the AOX models in that there are substrate non-substrate models, but we don't have sites of models. For the UGTs, we rely on smirk strings, which will identify potential sites of UGT metabolism on the molecule, and then compare the actual UGT isoforms to see if they metabolize one of those functional groups. So here I have 2 molecules. I'm going to go ahead and calculate ADMET properties and then go into the metabolism tab here. And we see we have UGT models here. So for the first one, it predicts only one of the 9 isoforms, UGT2B7, to metabolize this compound. So that's all fine. If we double click on this to bring it into MedChem Designer and then go under the Met button here and click on UGT, it will add a glucuronide to the OH here to form the glucuronidated product. The other compound, if we bring it into MedChem Designer by double clicking on it and then predict the UGT metabolites, here it's only predicted UGT 1A8, glucuronidate, the nitrogen here. And that's overriding the other model, 1A1. In this next section, I'll discuss our esterase models. These models are strictly transformations, so they're associated with the CRF file, which I'll show in a minute. But let's take a few examples here. So the first compound is a cyclovir, and it's a prodrug. All these are according to their Kimball data sets. And again, we can just double click on the compound to bring it up into MedChem Designer. And then I'll click on the esterase option under the Met button. And we see that the compound has been cleaved, the ester bond has been cleaved here. So this is a simple valine functional group that's been added to the compound, and then this is cleaved by an esterase to transform it into an active form. And the reason they would do this is because maybe this compound itself has low bioavailability, and so they would protect it with an ester to make it a little bit more orally bioavailable. This next example is for a phosphase containing compound. Again, I'll just click on the double click on the compound to bring it into MedChem Designer. And here it's cleaved this phosphate group off here to leave the hydroxyl group. Then this molecule is not an ester, it's actually a carbamate, but it can be cleaved in the same way. Now there's a couple different steps involved in this one. So esterases could cleave here to leave the OH here, and then cleave again at this particular bond to generate an unstable intermediate. Basically, when you have this N CO2 group, that's unstable and will just decompose into the amine. So let's see what happens here when we choose the esterate functionality. What it's done is it's really cleaved at this bond, which formed an unstable intermediate, and then released the amine here. And then finally, a bit more complicated analysis here. Now we have an ester group. Not sure if this gets cleaved or not. Let's see what the program does. So it does cleave this bond here to form the PO3H2 group here, which is the active molecule for tenofovir. Now let's take a brief look at the files that generate these metabolites. So in my user app data directory here, and you can see these three files here: metabolite prediction.crf, metabolite prediction esterase, and metabolite prediction UGT. Now this one is actually in an encrypted form, but you're certainly welcome to look at the compounds, or to look at the transformations here. And as I mentioned, they are encoded, but we do have comments for transforming these. If we could uncomment or unencrypt these, we'd see that on the left-hand side of the equation, there's an atom that is marked, I think, with an underscore S, and that indicates that it is the site of metabolism. And the program internally would mark it as a site of metabolism and then perform the transformation, which would generate the final product's metabolism. If we look at the esterase functionality here, or excuse me, the esterase CRF file, this is not commented, but it are not encrypted, but there are a large number of equations in here, or reactions, excuse me. So this is a SMERC string. It identifies the reacting functionality of the compound, and there can be wildcards. So for example, this could be nitrogen, oxygen, or sulfur, I believe here. And then this colon 3 marks that and tells it what it's going to be transformed into the product here. And then finally, the final one is the UGT one. And this one works in conjunction with other rules, but essentially marks the site of metabolism on the molecule and then adds the glucuronidate compound to it. In this last section of the video, I'd like to discuss metabolites and metabolites and generating metabolites within the batch functionality of ADMET Predictor. Here I've got a couple of simple molecules sketched into MedChem Designer, and I'm going to highlight one of the molecules and then just generate the standard cytochrome P450 metabolites of the compound. And you see this metabolite M2 here in this compound is a result of demethylation of the methoxy group here. And if I want to see if this potentially would be glucuronidated, I would highlight the compound and then go up into the MET button, select UGT, and we see that it glucuronidates the phenolic oxygen of the compound. So this is a metabolite of metabolite. Now let's go back to the other structure in MedChem Designer, and we're going to generate metabolites here. And then down in the lower left-hand corner, you see this is generated in aldehyde. Now, aldehydes are reactive compounds, so typically the body is going to try and get rid of that. So let's highlight this compound and again generate metabolites. And you see here that we're predicting that aldehyde oxidase has converted the aldehyde into a carboxylic acid. And that's one of the main functions of aldehyde oxidase and helps to detoxify the aldehyde group. I also should mention that CYP3A4 can also metabolize aldehydes into carboxylic acids. In the final section, I want to just describe the batch functionality within ADMET Predictors. You can highlight as many compounds as you want. Let's say we'll just highlight a few of these compounds. And then if I right-click in this area, we can go under Generate Metabolites. You have your options for the CYPs, UGT, and esterases. I'm going to generate CYP metabolites. And then within the GUI here, you can go through a number of metabolic cycles. So you could generate one cycle for all of these, and then if you increase it, it's going to generate metabolites of metabolites. So let's just perform that function very quickly here. It's going to open up a new ADMET Predictor spreadsheet with these metabolites, and I'll kind of widen this out. So here's the base compound. Then here's some metabolites of that base compound, and they're all going to have an M number associated with them. And then when you see an MM, that means it's a metabolite of a metabolite. So it's predicted the metabolites of this first compound kind of in the first run, and then a second metabolite of a metabolite. You can also generate maybe the first round of metabolites of CYP metabolites, and then expose this to the go on and generate metabolites maybe of UGTs for this particular compound, and that will generate in the first phase CYP metabolites, and then in the second phase the glucuronidated form of the compounds. Well, that's it for the metabolite generation video, and if you have any questions, you can email me. Thank you, and I'll see you next time. Bye-bye.